Hello students, in this video lecture I'll talk about classification of crude drugs. So here we go. The term crude drug generally applies to the parts or products of natural sources like plants, animals and minerals. They are found in raw and unprocessed form which are treated for their packing and to prevent them from deterioration. You can understand it with simple examples that are part of our routine like fennel, clove, coriander and amla. These are some crude drugs classification methods. First one is alphabetical classification, then morphological classification, pharmacological, chemical, taxonomical, and chemotaxonomical classification. Further, we will discuss them in detail, but before that, we should know why there is need to classify crude drugs. Although each system has its own merits and demerits. Let's take the example of a library. Libraries can vary in what methods they use to catalog and organize their collection, as it is easier to find our book of interest from organized one than from a messy library. Likewise, classification of crude drugs and the arrangement of classification in a definite sequence is a necessary to understand easily. So let's proceed to discuss them in sequence. First one is alphabetical classification, in which drugs are arranged in alphabetical order of their Latin and English names. You can see in different pharmacopoeias and reference books like Indian Pharmacopoeia, British Pharmacopoeia, United States Pharmacopoeia, and European Pharmacopoeia. Here you can see in the example, first two drugs, amla and agar, start with alphabet A, then benjoin and beeswax with B, and so on. The advantage of this method is that it is a simple method, as addition and identification of the drug is easy. And the disadvantage is that we can't find whether it is organized or unorganized, which we will discuss in next method of classification, that is morphological classification, in which drugs are classified on the basis of part or product of the drug source. So if we are talking about part, we call them organized drugs, as its name indicates. Organized means a particular organ is used which may have a particular shape or structure like leaf, bark, fruit, root. Likewise, products of plant, animal and mineral source are called unorganized drugs. For example, dried latex, dried extracts, gums. These are the examples of cellular or organized drugs because cells are present in them. Like fruit of amla, fennel and coriander is used as a source of drug, and bark of arjuna, cinchona and ashoka. You can note other examples also. And these are the examples of unorganized or acellular drugs because cells are absent in them. Dried latex of opium and papain used as a source of drug, and dried juice of aloe and quinoa. Advantage of this method is that it is convenient for practical study when chemical nature of the drug is not clearly understood, and disadvantage is we can't know the therapeutic action of a particular drug. To rectify this disadvantage, next method developed, which is pharmacological classification, in which drugs are categorized on the basis of pharmacological or therapeutic action of the main active constituent present in them. Like coriander, fennel, and clove are used as carminative, vinca and podophyllum as anti-cancer. Advantage of this system is that even if we don't know the chemical constituent present in the drug, its therapeutic action can be known, and disadvantage is we can know the chemical nature of the constituent to which therapeutic activity is attributed, which we can study in chemical classification, which was developed to rectify this problem or disadvantage. It is a preferred method of classification because in this both chemical nature and therapeutic efficacy can be known. These are the some examples. Belladonna, datura, and vinca are found to contain alkaloids. Glycosides are present in senna, cascara, and digitalis. As I told you earlier, every method has its demerits too, like in this. First is it is difficult to identify drugs containing two different constituents of equal importance. For example, fixed oil and volatile oil in nutmeg, alkaloids in glycosides in cinchona. Second is we can find the origin or the family to which they belong. So to rectify this, next method developed, which is taxonomical classification, in which drugs are arranged into phylum, order, family, genus, and species. Like we can see in these examples, monocot angiosperms, colchicum and asparagus belong to the same family, that is Liliaceae, and dicot angiosperms, Glycyrrhiza and senna belong to the Leguminosae. Later, equal importance was given to both taxonomical status and chemical constituents in chemotaxonomical classification, as you can see in the examples. Volatile oils present in members of both Umbelliferae and Rutaceae, and tropane alkaloids in the members of Solanaceae. You can note these questions as these type of questions can be asked in exams. This is all about this lecture. Thank you.